Hello, I'm Julie Theriault from Stanford University, and I am back for the third part of my iBioseminars presentation. Now, in the first two parts, I focused on well-understood systems with strong experimental evidence supporting all of the conclusions that I was telling you. For the third part, I'm going to venture into the field of wild speculation. And in fact, I'm going to wildly speculate about the biggest questions of all in the context of biology, which is how things evolved. And in particular, what I'd like to discuss is some big picture ideas about how the dynamic cytoskeleton has evolved throughout the history of life on Earth in order to give us such an incredibly diverse array of cell types, of structures, and of behaviors of cells. And just to illustrate that, here on the left I have an image of one of the most fabulous of protozoa called Stentor, which is an absolutely enormous cell with, as you see, a great deal of very detailed subcellular specialization in terms of its structure. And then on the right, a luridly colored electron micrograph of some bacteria, which are quite diverse in terms of their molecular components, but fairly similar and fairly simple in terms of their shape. Now, one of the things that's become very clear in the modern era of sequencing is confirming something that, of course, Darwin had originally speculated about quite some time ago, which is this proposition that all of the organisms that are currently living on our planet are descended from a common cellular ancestor or a common pool of cellular ancestors. And certainly one of the most undeniable pieces of evidence in support of that hypothesis is the observation that there's a tremendous number of molecular components that are found highly conserved throughout all branches of life. This particular illustration shows the sequence similarity among ribosomal RNA for bacteria, for archaea, and for eukaryotes. And the fact that it is possible to arrange all of the known living cells on Earth in this sort of tree where everything looks related to everything else is a very good indication that, in fact, all of the ribosomal RNAs that exist in cells on the modern Earth all came from some common ancestor back at some distant point in the past. That common ancestor on this diagram is indicated with the green circle, and it's fondly called LUCA, which stands for the last universal common ancestor. Now, this was not the first cell. This was just the last cell that gave rise to everything that's living today. And it was already very complex. It already had ribosomes. It already had DNA. It already had most of the components of central metabolism, including, for example, all of the enzymes involved in glycolysis. And it also had cytoskeletal components. And we know this because those common components are found in every single branch of currently living organisms. Now, given that everybody all started off as part of the same family, it is interesting, I think, to speculate about why it is that some parts of this tree of life have ended up behaving so differently from other parts of the tree of life. And in particular, I'd like to draw the distinction between what I will loosely call the prokaryotes, which are on the top part of the tree, and that includes the bacteria and the archaea, which are very distinct branches, versus the eukaryotes on the bottom part of the tree. Now, although bacteria and archaea are no more closely related to each other than they are to eukaryotes, they nonetheless are quite similar morphologically. And in fact, we didn't even know that the archaea existed as a separate kingdom until sequencing data started coming along. Because if you just look down a microscope, it's very hard to tell the difference between a bacterium and an archaeon. Whereas the morphology is very simple in those prokaryotes, the metabolism is extremely diverse. So almost any interesting chemistry that's done in living organisms was first invented by the prokaryotes. So what have we got going for us down here on the eukaryotic side of the tree? Well, although our chemistry is not necessarily as interesting or as complex as what's going on with the little guys, we have morphology. We get very big, we get very complicated, and we get very multicellular. So within this context of everybody being related to everybody else, and in fact, all of these different organisms having something that looks like cytoskeletal proteins, I think it becomes a very interesting question to ask, well, what is it really then that makes eukaryotes different from prokaryotes? Specifically, what is it that gives eukaryotes the ability to have such extremely diverse and complicated cellular morphologies based on cytoskeletal structures? Just to, again, give you a little bit of a taste of the kind of diversity you find even within a very closely related group of eukaryotes. These are some illustrations from one of the great microscopists of the 19th century, just showing a group of fairly closely related ciliates and flagellates. Each of these are unicellular organisms. They're not all drawn to scale with each other. This is Stentor that I showed you a photograph on the very first slide. and a number of other familiar organisms, including, for example, Paramecium here and Giardia. And all of these cells, to a first approximation, have the same components. If you look at the protein components of these cells, there certainly are things that are a little bit different between them, but the major components, certainly the major structural components, are pretty well conserved. And yet somehow all these different organisms are able to make drastically different cell shapes with very different motile behaviors, very different kinds of division behaviors, simply by rearranging this common set of molecular components. Now, if we contrast that to the bacteria, you certainly will hear proponents of the prokaryotic side of the tree claiming that there is a lot of morphological diversity among bacteria. And to a small extent, that's true. So if we look at this illustration from a really enchanting review by Kevin Young that came out a few years ago, this shows essentially the diversity of the different kinds of shapes that have been described for isolated bacterial species. And here in the zoomed in version, we find a couple of the fairly familiar shapes. So we see things like E. coli here, which is a rod shape. There's some crescent-shaped bacteria here like Colobacter. There are helical-shaped bacteria. But they're all sort of fairly simple variations on a theme. And then all of those guys are also quite small. And then we zoom out to higher magnification, there's really very few bacterial species that get much bigger than just a few microns in characteristic size. One interesting exception is illustrated here by this yellow dome that stretches across this drawing. And this is actually to scale with the other bacteria here, an illustration of an organism called Thiomargarita, which is one of the largest bacteria known. And these guys actually can be almost a millimeter across. And this shows a photograph of Thiomargarita next to a Drosophila. This is a single bacterial cell, and you can see it's absolutely enormous size. But the trick here is that even though the cell itself is very large, its cytoplasm is very small. Thiomargarita is basically just a hollow shell of a very thin layer, a couple microns thick of cytoplasm that's stretched around an enormous vacuole. So even when bacteria get to be pretty large and relative for bacteria morphologically complex, they still have diffusion-limited dimensions. And it is obviously observable that overall, if you take a random eukaryote and compare it to a random prokaryote, the eukaryotic cell is bigger and is much more structurally complex. So this just pulls out a couple of those things as examples. Here's Paramecium compared to a bacterium to scale, and then that bacterium blown up. And here I've chosen Vibrio cholerae. And you can see, again, there is subcellular localization, subcellular specialization in the context of Vibrio cholerae. It's got its flagellum growing out of one pole, for example. So it somehow is able to know which end is which and know the difference between the poles and the sides. But even at that level, the complexity is just vastly less than what you see in something like Paramecium. So if you think about what are all the observable differences between eukaryotes and prokaryotes, there's a pretty long list that is, there are some exceptions, but this is reasonably typical, reasonably representative of these different kinds of life. Eukaryotes, of course, have a membrane-enclosed nucleus. That's the definition. And so that means they have separated spatially and temporally the process of transcription from the process of translation, which gives rise to many options for very complicated regulation that basically can't be present in bacteria or archaea. Along with the membrane, the eukaryotes also, the nuclear membrane, the eukaryotes, of course, also have lots of other internal membranes, making things like the Golgi apparatus, the endoplasmic reticulum. They typically have much, much larger genomes than our bacterial friends, with often multiple chromosomes that can be very large. As I mentioned, they have much larger cell size. Instead of being characteristically just a few microns long, they're usually a few tens to even hundreds of microns in size. And they have a very high degree of subcellular compartmentalization. In addition, eukaryotes have endosymbionts, so they have mitochondria and chloroplasts that used to be bacteria, but have now been captured and domesticated by the eukaryotic cells in order to perform energy functions. And then finally, the reason we're even able to talk about this is because eukaryotes make multicellular organisms. Now again, just as in the context of the shape, it's not the case that bacteria never make multicellular organisms. It's just that they're not very complicated. So for example, the best understood multicellular bacterial organism is this thing called a stromatolite. We see a bunch of stromatolites here growing in an ocean next to Australia. And these are basically big piles of colonial cyanobacteria that grow and deposit some extracellular matrix, and then the next generation will grow on top of it, making these very characteristic layer cake patterns that are seen not only in the kinds of stromatolites we have growing today, but also are found in stromatolites that are several hundred billion years old in the fossil record. And in all that time, look how far eukaryotes have come, but bacteria are basically still making the same structures. Eukaryotes have been able to make things like redwood trees, they've been able to make things like coral reefs with this extraordinarily morphological diversification. Okay, so this very fundamental question of what is the difference between eukaryotes and prokaryotes that gives us such extraordinary morphological complexity is a question that I would say today we don't know the answer to. But a few decades ago, we thought we did know the answer to it. And before 1990 or so, the nearly universally accepted explanation for why there's such big morphological differences came down to the eukaryotic cytoskeleton. If we go through all of those characteristics I described about what's special about eukaryotes as compared to bacteria or archaea, all of those things can be attributed to functions of the cytoskeleton. So for example, the existence of the membrane-enclosed nucleus and all of the complex internal membrane systems can be ascribed as being due to intracellular membrane transport with motor proteins, for example, pulling vesicles along microtubules. Nuclear lamins, which are essentially a form of intermediate filaments, stabilize the nuclear membrane. The expanded genome in eukaryotes, of course, is divided by the extraordinary machine of the mitotic spindle, which with all of its self-organized microtubules and its massive amounts of flux and force generated by motor proteins such as myosin, or such as kinesins and dyneins, is able to divide a lot of chromosomes. And it's no big deal for a eukaryote to generate an extra chromosome, split a chromosome in half, vastly expand the size of its genome, because the mitotic spindle is able to handle a lot of capacity in terms of segregating chromosomes. The much larger cell size of eukaryotes can be attributed to the fact that they're no longer diffusion limited. Because of the directed action of motor proteins, you can actually have transport and mixing within the cytoplasmic compartment. Similarly, the subcellular compartmentalization, the existence of endosymbionts, and even the ability to have cells come together and make multicellular organisms with very complex extracellular matrices largely involves coupling between microtubules, actin, intermediate filaments, and other components within the cell or from one cell to another. Endosymbionts, for example, are thought to be the product of a phagocytic event where an ancient eukaryotic ancestor ate a cyanobacterium and turned it into a chloroplast. Now, the reason I say we don't know the answer today, even though we did know the answer in 1990, was because it became clear in the 1990s that bacteria actually do have a cytoskeleton that in many ways is remarkably similar to that of eukaryotes. And so the first real nail in this coffin was the discovery of a tubulin homologue in bacteria that goes by the name of FTSZ. FTSZ had been originally identified genetically as being a protein that was required for cell division. And some beautiful work in the 1990s combining biochemistry and cell biology of this protein has established that it really acts as a legitimate cytoskeletal protein in bacteria that's involved in cell division. It forms a ring right around the middle of a bacterium that's about to divide, and as the bacterium divides, that ring's actually becomes smaller. Furthermore, the purified protein is able to self-assemble into filaments in a manner that depends on the presence of hydrolyzable GTP, which again is a very cytoskeletal behavior. And any remaining doubt had to be squashed when the crystal structures came out for both tubulin and FTSZ at about the same time, back to back in the same issue of Nature. And it was clear that the structure of one could be almost perfectly superimposed on the structure of the other. So it's clear that these are not only sort of analogous proteins, but in fact true homologues in the sense that they are descended from a common ancestor. And by far the simplest explanation for that is that the LUCA, the last universal common ancestor, also had a protein like tubulin or FTSZ. Okay, so bacteria have something like tubulin. Then it turned out a couple years later, bacteria also have something like actin. And just like with FTSZ, these proteins that go by several names, there's MRAB and MBL and a few other family members, were originally described as being genes that are necessary for some aspect of bacterial shape, a fundamental cytoskeletal type function. And so for example, in Bacillus subtilis, disruption of any of these actin homologues causes really abnormal overall types of cell shapes. And these proteins, when purified, can assemble into filaments, again like FTSZ or like any of the eukaryotic cytoskeletal proteins. Now, one thing that's been, I think, particularly amusing in the context of the bacterial actin homologues has been the proliferation of such a large number of them that appear to have slightly different specialized functions. So for example, one of my favorite cases is in magnetotactic bacteria that are able to orient themselves with respect to the Earth's magnetic field. They do so by lining up these little crystals of magnetite along a structural element inside the cell that is in fact made up of an actin-like filament. Now this is not the same actin-like filament that is contributing to cell shape. It's actually a copy of the gene that was historically duplicated and then diverged for this specialized function. So bacteria not only have actin, they have a bunch of different actins, but they can use them both to determine their overall shape and to determine the distribution of intracellular organelles. So now I think we're set up to ask ourselves a slightly different question. It's clear that bacteria do have a cytoskeleton. So the existence of the cytoskeleton cannot be the thing that distinguishes eukaryotes from prokaryotes. So we might ask then, well, if bacteria do have a cytoskeleton, then why don't they do something more interesting with it? And by that, I mean in this very eukaryotic-centric worldview, why don't they have morphological complexity? And why don't they make big, fancy multicellular organisms? So here we get to the point of wild speculation. And my specific hypothesis is that the thing that all of the cytoskeletons have in common is the ability to perform large-scale cell organization based on self-assembly of helical filaments that are highly dynamic. That's true among all the different branches of life. I am proposing that what is special about eukaryotes is not the cytoskeletal filaments per se, but rather two specific classes of cytoskeletal associated proteins, the nucleators and the molecular motors. And I'll go into some detail about why I think these are the things most likely to really be different. But so far, there is no strong positive evidence that prokaryotes have any classical molecular motor proteins or any regulated nucleators for cytoskeletal filaments. Which raises a second question, if the prokaryotes don't have them, why don't they have them? That's something even I'm not going to be able to speculate about. But as far as why I think these two things are really the key to this morphological difference, I'll give you some of the information that has led me to that proposition and suggest a few very specific ways that these ideas could be tested. Okay, so the overall basis for this hypothesis that you need nucleators and molecular motor proteins in order to get morphological diversity comes from a series of observations about the kinds of structures that can be made by cytoskeletal filaments in bacteria versus in eukaryotes. In particular, any self-assembling helical filament can make either a structure with a single filament or can make a bundle or a raft of filaments where the filaments are oriented in random directions with respect to each other. All that requires is helical self-assembly, which, as we'll see, is a very universal feature of proteins that are able to interact with themselves. On the other hand, the kinds of structures that really require localized nucleation or localized motor activity are illustrated here on the bottom. These include things like microtubule asters, which are familiar from the organization of overall organelles inside of a cell. These include things like parallel bundles, which we find inside of eukaryotic flagella, or we find in muscle, the sarcomeres that are able to give large-scale muscle contraction. And also things that have bipolar morphology, like the mitotic spindle shown here. We know enough about how these different structures are formed that I feel fairly confident in stating that the easiest way to make these kinds of structures is either by localizing nucleation of filament growth or having molecular motor proteins that are able to sort out filaments of different orientations, or some combination of both things. And what I will assert is that no structures of the type shown under here, class B, have yet been found in bacteria, specifically have yet been found in the cytoplasm of bacteria, with perhaps one or two interesting exceptions that I think actually sort of prove this rule. So if nucleators and molecular motor proteins essentially enable you to take your helical, self-polymerizing, self-assembling cytoskeletal filaments and make large structures out of them, you might ask, well, what evidence is there that eukaryotes can do this but prokaryotes cannot? So I'd like to now again take a step back and say, if we are willing to accept the premise that something about the activity of the eukaryotic cytoskeleton is the key difference, then the question is, what is special about the eukaryotic cytoskeleton? And if you look at the most abundant and most studied eukaryotic cytoskeletal proteins, we have tubulin making microtubules, actin monomers making actin filaments, and then intermediate filaments made up of their own subunits. And what you immediately see looking at all these things is that they're all basically helical self-assembled structures. But I would argue that helicity of a self-assembled structure is not something that's particularly difficult to evolve. And in fact, there was a very strong argument made first by Crane and then by Pauling back in the 1950s that pretty much any protein that has any tendency to associate with itself is more likely to form a helix than it is to form anything else. And the argument's pretty simple. If you have a globular protein where every aspect of that protein surface has slightly different physical attributes, slightly different charge distribution, slightly different shape, and that protein has some tendency to interact with a second copy of itself in some particular orientation where, part A of one protein binds to part B of another protein. If the protein is going to make a dimer, those two things that interact with each other have to be distributed symmetrically on the protein. But if the part A and the part B are in any orientation other than just directly opposite each other, then essentially what's going to happen is those two subunits will come together and there will be an interaction between the two that will give a dimer that's got some slightly off-center asymmetric structure where if part A and part B are engaged here, this subunit still has a part B available and this subunit still has a part A available. Now, if you think that through, as these guys did, what you see is that you can then add another subunit on this side, you can add another subunit on that side, and you're able to make helices out of this very simple binary interaction rule. And in this really creative and beautiful illustration done in this original paper using little matchboxes, you can see you can actually build all sorts of different kinds of helices just by slightly changing the orientation of the interaction between those two subunits. So making a helix is not a big deal. And in fact, a really great piece of evidence to show that that's true is the effect of the sickle cell mutation on hemoglobin. Now, hemoglobin is highly selected to be very, very soluble. In red blood cells, the concentration of hemoglobin can get up to on the order of 300 mg per mil. But just one point mutation in hemoglobin can give it the tendency to precipitate. And when it precipitates, it forms these incredible-looking helical filaments that look really almost exactly like microtubules. If I told you this was a microtubule, you would probably even believe me. But it's actually a polymer of sickle cell hemoglobin. So when you take a very soluble protein and you make something just go a little bit wrong with one part of its surface so that it has a very minor tendency to self-associate, the consequence is you get a helix. Okay, so forming a helix is not a special property. What is it about cytoskeletal filaments that gives them their ability to give us morphology? Well, one other important thing that is true of all of the eukaryotic cytoskeletal filaments is that they have this very interesting dichotomy that they have to be stable in order to have physical strength, but they also have to be unstable in order to allow the cell to change shape or to move or to rearrange its elements as it's dividing. And at least for actin and for tubulin, part of the reason that they're able to exhibit this dichotomy is because those subunits are also able to bind and hydrolyze nucleotide. ATP in the case of actin, GTP in the case of tubulin. And as you see in this illustration here, the fact that you can have different conformational states of a protein based on the identity of the nucleotide that's bound to it can set up a situation where you can have one conformational state, in this case illustrated as being the thing binding ATP, which is a conformation appropriate for forming one of these helical filaments. And you can have an alternative conformational state, which is shown here in this case as being the nucleotide-free state, but it could also be the ADP state, that will tend to disassemble. And so having that switch between an assembly-competent form and an assembly-incompetent form can enable the dynamics that are characteristic of eukaryotic cytoskeletal filaments. And in fact, it's very interesting that actin itself is a close structural relative of hexokinase, the glycolytic enzyme, which is best characterized with respect to its very large conformational change on binding and hydrolysis of substrate. And in fact, it's true that if you look at the cytoskeletal elements of eukaryotic cells, they're all highly dynamic. And this is something that's been appreciated for many decades in the context of microtubules, actin, and intermediate filaments. All of them turn over very fast inside of cells. And in fact, for things that require the cytoskeletal filaments, like for example, forming a mitotic spindle, if you inhibit either the assembly or the disassembly of the microtubules in a mitotic spindle, the net effect is the same in both cases, which is that the cell fails to divide. So it's not just the helical structure, but also the turnover is clearly key. Okay, well, let's turn back to the prokaryotes. Do they have dynamics associated with their cytoskeletal filaments? Well, actually, it turns out they do. So as I mentioned, FTSZ, that's a close structural homolog of tubulin, also binds and hydrolyzes GTP, and in vivo turns over very, very rapidly, as can be seen in this photobleaching study. So here there are two cells that have assembled FTSZ rings. Here, 1/2 of one of those rings has been bleached out, and you can see actually within just a few seconds, the ring is able to come back. And this particular experiment also had a really, really nice direct demonstration that those dynamics are associated with nucleotide hydrolysis, which was they were able to compare the dynamics of wild-type FTSZ protein with an FTSZ protein that had a point mutation that slowed down GTP hydrolysis. And looking at the turnover of the wild-type protein versus the turnover of the slower protein in vivo, they were able to see that there's this direct correlation between how fast the filaments turn over and their ability to hydrolyze GTP. Okay, so the prokaryotic filaments, they do the same kinds of dynamics as the eukaryotic ones do. So it's not the helical structure, it's not the dynamics in terms of turnover, and it's not even fancy dynamics. So microtubules, for example, famously do this dynamic instability, where even in a uniform chemical environment, an individual microtubule end will grow for a while and then shrink for a while and then grow for a while and then shrink for a while. And it's not at all obvious why this should be possible. This is something that people have been studying for many years, and we have a pretty good overall chemical understanding of how this can happen. But it's definitely dependent on the ability of the structures to hydrolyze nucleotide and change their conformational states. So this illustration up top shows the classic direct demonstration of this dynamic instability on microtubules that was done in 1986. And then almost 20 years later, another group was able to show with one of the bacterial actin homologs, this one called PAR-M, that they're able to see exactly the same kinds of behaviors of these filaments growing for a while and then shrinking for a while, even when the chemical environment is uniform. Okay, so it's not the helicity, it's not the ability to hydrolyze nucleotide, it's not the ability to turn over rapidly in vivo, and it's not even the ability to do fancy things like dynamic instability. What is it then that's different? Well, looking at bacterial cells, it's clear that the way they're able to get some sort of overall cellular organization definitely depends on this tendency of proteins to make helical filaments. And in fact, if you look at bacteria and you look at different aspects of their structures, what you see is the helices are all over the place. And you can make lots of different kinds of helices, ranging from things like the overall helical pattern of a spirochete to the twisting helices associated with bacterial flagella to even the much more sort of uniform helix associated with something like a bacterial pilus. So it's clear it's not just the classical cytoskeletal proteins in bacteria that are able to do this helical self-assembly trick, but in fact lots of other proteins as well. And looking specifically at the cytoskeletal proteins, I think one interesting example is the bacterium Caulobacter crescentus, which is this very cute little banana shape with structural differentiation on its two ends. It's got, in fact, not just actin and tubulin, but it also has something that looks very much like an intermediate filament. And each of those things assumes slightly different helical patterns inside of a growing cell. And mutations in either the actin homolog, the tubulin homolog, or the thing that looks like an intermediate filament will affect the overall shape of the cell in very different ways. So lots of helical organization, lots of function associated with helical organization, but still no great morphological diversification. Now, like everything else in biology, there's going to be some exceptions. And there are some bacteria that have really particularly spectacular shapes. And this just shows a couple of my favorites. This is a star-shaped bacterium called Stella humosa. It's flat, but it has either 5 or 6 points, depending on what stage of its cell cycle it's at. This is another weird-looking flat prokaryote that in this case is an archaeon. This is called Haloquadratum walsbyi, and it was first cultured in pure form only a few years ago. And this is actually flat like a floor tile. And when it divides across the middle and then again like that. So you have one floor tile going into 4 smaller ones. And then there are things like this. This is Epulepicium fischelsonii, which competes with Thiomargarita as being one of the biggest bacteria known. And this is absolutely huge. It can be half a micron long, and it has many copies of its genome that are actually distributed in a very regular pattern throughout the entire cell. And it moves its genome around depending on what time of day it is. So I think for things like this, there's got to be something else going on besides just our usual bacterial helical self-organization. And I think it's an interesting challenge to now try to go into these organisms and find out what it is that's determining their ability to have these very specific morphologies. Okay, but getting back to the main thread, I've been arguing that we can have simple helical structures without motor proteins or nucleators. And in order to have more complicated things that make eukaryotes what they are, we need to have either nucleators or motors. So this raises the question, well, why don't bacteria have them? Where did they come from in eukaryotes? And why does it seem like it's so hard for the bacteria to pick them up? Well, for the nucleators, this is a particularly interesting problem because for the way that eukaryotic cells tend to nucleate their cytoskeletal proteins is very often by taking the subunit, copying that gene, and then having it diverge a little bit to assume a specialized nucleation function. And we've seen this in two cases, completely independent cases, for actin nucleation and for tubulin nucleation. So actin nucleation can be performed by this complex that's called the ARP23 complex, where ARP stands for actin-related protein, because there are two different proteins from different genes, both structural homologs of actin, that come together to make this regulated nucleation complex. In the case of microtubules, the nucleation is often carried out by what's called the gamma-tubulin ring complex. Now, the lattice of the microtubule is made up with alpha-tubulin and beta-tubulin. Gamma-tubulin is specialized only for nucleation function. Why can't bacteria do this? Well, I think they clearly could if they wanted to, because we do see, as I mentioned, among the actin homologs in bacteria, an individual cell may have actually several different kinds of actin homologs that are all present within that same genome, that have all been evolved for slightly different functions. And yet, so far as we know, none of them have been specialized specifically for filament nucleation. I don't know why they don't want to, but it seems like they could, and yet they don't. Turning down to the molecular motor proteins, this is something where it might be a little more clear why bacteria don't have them. If we think about where the really good molecular motor proteins come from in eukaryotes, there are, of course, three different classes. There's the myosins, the kinesins, and the dyneins that all hydrolyze nucleotide in order to undergo a conformational change. And of these, the kinesins and the dyneins move on microtubules, and the myosins move on actin filaments. Now, these different motor proteins had all been purified, had all been studied biochemically and sequenced, and then eventually their structures were determined. And at the point when the structures were determined, there was this huge surprise, and this again was back in the 1990s, where it was found that the fundamental structure of the catalytic core of myosin was almost identical to the structure of the catalytic core of kinesin. Even though myosin walks on actin filaments and kinesin walks on microtubules, they still seem to be homologs derived from a common ancestor. And in both cases, the fundamental way that ATP hydrolysis at the core of the motor protein results in the conformational change that gives you a step is very similar at the heart of the motor protein, even though all the details about how it couples to its filaments are quite different between the two. So this gives rise to another field ripe for speculation about where do the motor proteins come from. Well, if kinesins and myosins are related to each other, it seems like there must have been some sort of motor precursor. And we don't know what its substrate was. If it was actin, if it was microtubules, if it might have been something else. We don't know what the complement of motors was in the earliest thing that became a eukaryote, and whether this might have been something that actually drove it to separation from the prokaryotic branches of the tree in terms of morphological diversification. One thing we do know, though, is that those motor proteins, kinesin and myosin, are both derived from a particular branch of the superfamily of proteins that's called P-loop NTPases. It includes a lot of ATPases and GTPases. And this fairly complicated diagram is a summary of sequence diversification among many, many different family members of this large protein superfamily across the entire tree of life. And what this group, this is Eugene Koonen's group that's done this, has attempted to do is look at what the distribution is of all these protein types in currently living cells, and then project backwards in time to see when they might have evolved. And so there's some classes of proteins that you see here with these pink lines that are present in bacteria, in archaea, and in eukaryotes. And so they must have been present in LUCA, in the last universal common ancestor. But there's one particular protein family right here that's shown in brown that has both the myosins and kinesins in it, and then also has a whole lot of other GTPases that we associate with specifically eukaryotic functions. RAS, the Rab proteins that are involved in membrane trafficking, the Rho proteins that are involved in large-scale organization of cell polarity. All of those things come from this same relatively narrow branch of the P-loop NTPases. So one possible explanation for why eukaryotes and prokaryotes are different from one another is because the proteins that are best poised to become stepper motors of the kind that are familiar from eukaryotes happen to be in this branch of proteins that didn't evolve until the eukaryotic ancestor had already split off. It's very tempting to speculate that this particular class of proteins that includes not only the myosins and kinesins, but also all of these regulatory GTPases might be part of what makes eukaryotes what we are. Now, I'd like to point out, of course, there's plenty of other protein families that are not shared between eukaryotes and prokaryotes. About 50 other classes of proteins have been identified to date. However, I think mechanistically, this is a particularly intriguing group for generating specifically morphological diversification at the level of cells and at the level of whole organisms. Now, I don't mean to say that bacteria don't have motors. Bacteria have amazing motors. Bacteria have the flagellar rotor, which is a very complicated structure. It's much more complicated even than dynein. That's able to spin at 200 hertz and is made up of more than 40 different gene products. Bacteria also have extremely strong motors, like the motor that drives twitching by retraction of these type 4 pili. And this is something where a single motor interacting with filament that's extended from that's extended through the bacterial envelope is able to generate up to 100 piconewtons of force just from movement of a single extended filament structure. So the motors in bacteria can be very powerful, very efficient, and very complicated. And yet, all of them seem to do things on the surface of the bacterium. None of them seem to do anything in the cytoplasm. So somehow this question of morphological diversification about how you get complex intracellular structures seems to be at least correlated with the presence of linear stepper motors for cytoskeletal proteins in eukaryotes and no real equivalent in prokaryotes. And again, I can speculate wildly about why there seems to be this difference. But nevertheless, this is a fairly compelling correlation. Doesn't come down to complexity or anything like that, but really only down to the presence or absence of this one particular protein family. Okay, so this is overall the hypothesis again that we need nucleators and molecular motors in order to derive things like the mitotic spindle or like microtubule asters or like parallel bundles of cytoskeletal filaments that give eukaryotes morphological complexity. And I've made the claim that no structures of this kind, again with just very minor exceptions, have yet been found in the cytoplasmic compartment of bacteria. I'm focused, of course, on the role of the cytoskeleton in all of this, but it is worth thinking about maybe other kinds of explanations for the difference. And one thing that might really be connected is the fact that it's been observed in bacteria, where, of course, the chromosome is just embedded in the cytoplasm, and there's no nuclear membrane separating the chromosome from the cytoplasm. In the cases where it's been well documented, the bacterial chromosome is actually highly organized. One of the first hints of that came from these studies in Caulobacter, showing that a circular bacterial chromosome that was marked with different fluorophores at specific locations would actually organize itself in basically every cell within a bacterial colony such that those markers in the chromosome are all found in the same order. In other words, the bacterial chromosome is packed into the cell in a highly structured, highly regular way that can provide spatial information for targeting other kinds of subcellular structures, for example, to the poles or to the nascent septal zone. So, one possible way to bring these two ideas together is that at some point in the development of the eukaryotic ancestor, something happened that either enabled the nuclear envelope to come in or somehow otherwise separated the chromosome from the cytoplasm. And if bacterial cells and archaeal cells primarily rely on their chromosome as being their major organizing principle as far as where to put things in the cytoplasm, then when that separation happened, the little filaments that were left out in the cytoplasm that in bacteria had only been doing fairly trivial things of figuring out where to divide and how to determine the overall shape of where to lay down the cell wall, those guys were left by themselves out in the cytoplasm. They had no landmarks, they had no chromosome to tell them where anything was, and so they somehow had to figure out how to make larger scale structures like asters, like bundles, like spindles that could help them organize themselves in the cytoplasm in the absence of information from the chromosome. Okay, like I said, this was wild speculation. But one of the things I think is fun about this is that it's wild speculation that is absolutely disprovable. So I'd like to give you a personal challenge to look for and try to find either a cytoskeletal stepper motor that walks on a bacterial cytoskeletal filament in a way analogous to what kinesin or myosin does in eukaryotic cells, or else to find a regulated nucleator of any of these bacterial cytoskeletal filaments. A lot of very smart people have been looking in various ways for at least the past 20 years, and as far as I'm aware, nobody has yet found any proteins in one of these classes. Now, that certainly doesn't mean they're not out there, but it does mean that if either of these things were found, that would be absolutely definitive proof that my big crazy hypothesis is wrong. So please try to prove me wrong. And if you do find one of these classes of proteins, a bacterial cytoskeletal stepper motor or a bacterial regulated nucleator, please send me an email and let me know. I would love to hear about it. And if you do, I personally will send you a large bouquet of flowers and also hearty congratulations for taking another step forward to trying to understand one of these biggest questions about why cells on Earth look so different from one another. Thank you.